The fossil record is often presented as a neatly organized archive of life, where each discovery fits cleanly into an evolutionary tree, branching logically from one form to another. But this image is misleading. Scattered throughout Earth's deep past are creatures that do not fit. Organisms preserved in stone with no clear descendants, no obvious ancestors, and anatomical features so strange that even modern imaging and molecular analysis struggle to place them. These are not monsters born of myth or exaggeration. They are real animals, excavated from rock layers millions of years old, studied by professional paleontologists, and debated in scientific journals for decades. Some appear to combine traits from entirely different branches of life. Others seem to represent evolutionary experiments that vanished without explanation. This documentary explores those creatures alone, not legends, not speculation, but the prehistoric organisms that science has uncovered and still cannot fully explain. The Tully monster, the Carboniferous enigma. Beneath the warm, shallow waters of the Carboniferous seas, where sprawling tidal flats met vast swamp forests, an animal of bewildering design glided silently through murky currents. The Tully monster, Tully monstrum gregarium, thrived in an ecosystem already rich with life, yet it bore a form that seemed almost alien to the familiar blueprint of animal anatomy. Its elongated, soft body stretched more than 30 centimeters, tapering toward a rear fin that hints at a subtle, graceful propulsion. Unlike the armored or scaled creatures around it, the Tully monster relied entirely on soft tissues, captured in remarkable detail only through the unique preservational conditions of iron-rich shale deposits. A long, flexible proboscis projected from its head, ending in a pincer-like claw lined with sharp, pointed structures. This extraordinary feeding appendage has no parallel in modern or extinct animals. The claw's lateral movement suggests it could grasp or secure prey with precision, while the lack of a true jaw or skeletal support indicates a feeding strategy both delicate and specialized. In life, this organ would have allowed the Tully monster to extract soft-bodied organisms from the sediment or snatch prey from the water column, a method of hunting that defies simple categorization. On either side of its head, rigid stalks bore round eyes, positioned far apart to offer an expansive field of view. The placement and structure of these eyes suggest that vision was a primary sense for this predator, critical for detecting both prey and potential threats. Fossil evidence shows details in the eye tissue reminiscent of structures in vertebrates, although interpretations remain contested. Their unusual configuration reflects an evolutionary solution unlike anything seen in other Carboniferous animals, emphasizing that even sensory organs could diverge wildly from known forms. Running the length of the body, a faint internal band has sparked decades of debate. Some researchers interpret it as a notochord, a flexible internal support characteristic of primitive vertebrates, while others argue it represents a distortion or a preserved muscle structure. Near the head, paired features, interpreted as gill pouches, add another layer of complexity. If these elements are truly analogous to vertebrate anatomy, the Tully monster occupies a unique branch of early chordates. If not, it may represent a lineage entirely outside conventional classification. No current model satisfies all observed anatomical traits. Fossils of the Tully monster are surprisingly abundant, with thousands recovered from the Mazon Creek deposits in Illinois. Despite the wealth of specimens, no single fossil has provided a conclusive solution to its identity. Each example preserves some details clearly, while leaving others ambiguous, reinforcing the creature's status as an evolutionary outlier. Its consistent body plan across specimens confirms that these unusual features were not anomalies but defining characteristics of the species. Its habitat, a dynamic mix of brackish coastal waters and tidal channels, demanded adaptability. The Tully monster's streamlined body and wide field of vision imply a predatory lifestyle that relied on stealth and agility rather than brute force. Small crustaceans, juvenile fish, and other soft-bodied prey likely constituted its diet, captured with its delicate yet effective proboscis. Without natural armour or spines, it would have depended on awareness and swift, precise movement to survive among larger predators and competitors. What makes the Tully monster especially compelling is the abrupt end of its lineage. It vanishes from the fossil record without leaving transitional forms or descendants, a ghost lineage that reminds paleontologists how experimental evolution can be. This disappearance highlights the fragility of unique body plans in the face of environmental shifts and the relentless pressures of competition. Evolution produced a creature at once highly specialized and entirely ephemeral. Modern analysis continues to illuminate its mysteries. Advanced imaging has revealed subtle details in eye structures, soft tissues, and possible internal organs, yet the interpretations often conflict. Every new study brings insights and questions in equal measure, reinforcing the Tully monster's role as one of the most enigmatic animals of the Carboniferous. Its body plan, sensory apparatus, and feeding strategies illustrate the creative breadth of evolution, demonstrating that life has repeatedly explored solutions that leave no enduring legacy. Amid the shale of Illinois, the Tully monster lies preserved in stone, a silent testament to the ingenuity and unpredictability of evolution. It challenges assumptions about classification, sensory adaptation, and predation in early complex life. Even after decades of study, it resists the urge to be neatly categorized, standing as a vivid reminder that the history of life includes experiments both extraordinary and fleeting. In the shallow waters it once navigated, the Tully monster moved with silent efficiency, a predator unlike any other, now frozen forever in the record of time. Anomalocaris, the first giant predator. 500 million years ago, the oceans were teeming with life in forms barely recognizable to modern eyes. Among the myriad creatures of the Cambrian seas, one predator stood out, not only for its size, but for the sophistication of its hunting adaptations. Anomalocaris, a name meaning abnormal shrimp, was the first true apex predator, moving with precision and speed in waters crowded with small, soft-bodied organisms. Measuring up to 1 meter in length, it dominated the shallow seas, its body a marvel of evolutionary engineering, long before the appearance of fish or cephalopods. The reconstruction of Anomalocaris was not immediate. For decades, paleontologists misclassified its fossils, piecing together mouth parts, segmented lobes, and appendages as separate organisms. One fossil resembled a circular, tooth-lined disc, another suggested jointed limbs, yet another appeared as a leaf-like structure. It took decades of careful study to realize that all these fragments belonged to a single animal. This revelation transformed the understanding of Cambrian ecosystems and underscored the complexity that had emerged so early in animal evolution. The predator's body was elongated and flexible, with lateral lobes running along its sides that undulated in the water, propelling it forward with smooth, wave-like strokes. This mode of swimming allowed Anomalocaris to maneuver with remarkable agility, darting after prey or hovering near the seafloor to strike. Its streamlined form, paired with a flexible body plan, gave it a speed and grace unparalleled among its contemporaries. Even in a world dominated by soft-bodied animals, Anomalocaris was a formidable hunter. At the front of its head, a pair of jointed, spiny appendages reached forward like grasping arms. Each segment bore sharp, curved spines, perfectly suited for seizing and holding small prey. The appendages could bend inward, forming a claw-like mechanism, while the mouth, a circular, plate-lined oral cone, allowed it to consume soft-bodied animals efficiently. Fossil evidence suggests that while Anomalocaris could crush lightly armoured organisms, it specialised in soft prey, using both precision and speed to capture what other predators could not. Perhaps most extraordinary were its eyes. Mounted on stalks and composed of thousands of lenses, they were among the most advanced visual organs of the Cambrian period. Each compound eye could detect subtle movements across a wide field, an adaptation that gave Anomalocaris a decisive advantage in hunting. The combination of sophisticated vision, agile appendages, and a flexible body made it one of the first predators capable of actively pursuing its prey rather than relying on ambush or filter feeding. Anomalocaris's presence reshaped the ecological landscape. Its emergence drove the evolution of defensive adaptations in other animals. Small creatures developed hardened shells, spines, and burrowing strategies, responding to the constant threat posed by this predator. In this sense, Anomalocaris was not just a hunter, but a catalyst for evolutionary innovation, an early agent of natural selection that forced entire communities to adapt or perish. Despite its dominance, the lineage of Anomalocaris eventually disappeared. By the late Cambrian, more specialized predators and efficient arthropods had begun to occupy similar ecological niches, leaving Anomalocaris and its relatives increasingly marginalized. Fossils reveal a decline in size and abundance, marking the end of a pioneering chapter in the history of predation. Yet its legacy endures in the evolutionary experiments it inspired, evident in the more specialized predatory forms that would follow. Fossil evidence continues to provide remarkable insights. The Burgess Shale and Chengjiang deposits reveal not only the form of Anomalocaris, but also clues to its behavior and lifestyle. Impressions of appendages suggest how it grasped prey, while the body's flexibility hints at precise maneuvering in three-dimensional water space. Each fossil tells a story of a predator perfectly adapted to its environment, combining speed, vision, and grasping ability in a way that would not be repeated for millions of years. Anomalocaris reminds us that complexity arose early in life's history. The Cambrian explosion was not a gradual accumulation of simple forms, but a sudden bloom of diverse, specialized animals, each exploring unique solutions to the challenges of survival. Among them, Anomalocaris stands as the first true giant predator, an embodiment of evolutionary innovation, predation, and ecological dominance, a creature that, for a brief moment in deep time, ruled the seas with precision and power. Opabinia, the five-eyed enigma. The Cambrian seas were a world of experimentation, a time when the oceans became laboratories for life, to explore forms and functions with abandon. Among the many creatures that emerged during this period, Opabinia regalis stands out as one of the most extraordinary. Measuring only a few centimetres in length, this soft-bodied animal was a marvel of design, with 5 stalked eyes, a flexible proboscis ending in a grasping claw, and lateral lobes that allowed it to glide effortlessly through the water. Its body defied categorization, presenting scientists with one of the greatest puzzles of early animal evolution. Opabinia's 5 eyes immediately capture attention. Positioned along the top of the head, these stalked structures provided nearly panoramic vision, a rare adaptation in any ecosystem. The arrangement 2 on the sides, 2 nearer the center, and 1 on the top allowed Opabinia to detect predators and prey across a broad range of angles. In the dim light of Cambrian waters, such a sensory advantage would have been crucial. Each eye, composed of numerous lenses, hints at a visual sophistication that rivals many modern arthropods, suggesting that even small animals in these early seas relied heavily on sight for survival. Extending from the front of the head was a long, tubular proboscis, ending in a claw-like appendage. Fossil evidence indicates this organ was flexible and capable of precise movements, allowing Opabinia to probe crevices or grasp individual prey items. Unlike Anomalocaris, which pursued its prey with speed, Opabinia appears adapted for a more deliberate style of hunting, sweeping its proboscis across the seafloor to capture soft-bodied organisms. This unique feeding apparatus is unlike anything seen in later animal lineages, emphasizing the experimental nature of Cambrian body plans. The body itself was segmented and laterally flattened, with overlapping lobes along each side that acted as swimming flaps. These lobes created an undulating motion through the water, providing both stability and propulsion. Fossil impressions reveal delicate, gill-like structures beneath the flaps, suggesting that Opabinia combined locomotion and respiration in a single functional system. Its small size and flexible form allowed it to hover above the seafloor or navigate tight spaces in search of food, exploiting ecological niches unavailable to larger predators. Opabinia's place in the evolutionary tree has long been debated. Early scientists attempted to align it with arthropods due to its segmented body and lateral appendages, but it lacks the jointed limbs and hard exoskeleton characteristic of true arthropods. Its 5 eyes and unusual proboscis also set it apart from annelid worms and other known groups. Today, Opabinia is recognized as a stem group organism, a relative of arthropods that experiments with anatomical solutions ultimately abandoned by evolution. Its oddity highlights the breadth of innovation during the Cambrian explosion, a period when natural selection explored forms that would never reappear. Fossil evidence from the Burgess Shale captures Opabinia in remarkable detail. The preservation of soft tissues allows paleontologists to reconstruct not only its anatomy, but also its likely behaviors. Its undulating swimming motion, paired with precise manipulation of the proboscis, suggests a predator adapted for careful hunting rather than open pursuit. Despite its small size, Opabinia's unique sensory and feeding adaptations gave it a distinct advantage in a competitive marine environment, allowing it to occupy an ecological niche unfilled by other animals. Opabinia's extinction, like that of many Cambrian organisms, remains a mystery. Its disappearance may have resulted from increasing competition, environmental changes, or the rise of more efficient predators. Whatever the cause, Opabinia left no descendants, and its unusual body plan vanished from the seas. Yet it remains a symbol of evolution's capacity for experimentation, a fleeting but extraordinary success that demonstrates the creativity of life during its earliest complex chapters. In examining Opabinia, scientists are reminded that evolutionary success does not always follow familiar patterns. Some creatures thrive by innovation, filling niches with precision and ingenuity, only to vanish when conditions change. The 5-eyed predator of the Cambrian seas offers a window into a world of experimentation, a time when life pushed the boundaries of what an animal could be and left behind forms that challenge the imagination of modern scientists. Hallucigenia, the walking nightmare of the Cambrian. In the shallow seas of the Cambrian period, over 500 million years ago, life was experimenting with forms that seem almost alien to modern eyes. Among these early creatures, Hallucigenia sparsa stands out as one of the most extraordinary and perplexing. Its fossilized remains, first discovered in the Burgess Shale of Canada, have challenged scientists for decades. With its tubular body, paired spines along the back, and a row of leg-like appendages that defy conventional interpretation, Hallucigenia looks less like an ancestor of modern animals and more like a living nightmare. Yet, within this surreal design lies a remarkable story of adaptation, survival, and evolutionary ingenuity. Hallucigenia measured only a few centimeters in length, yet every aspect of its anatomy reflects a precise adaptation to its environment. Fossils reveal a soft, elongated body covered with rigid spines protruding from its dorsal side. These spines, once thought to be legs by early researchers, served as protective armor against predators, deterring larger Cambrian hunters such as Anomalocaris. Their sheer size relative to the body would have made the creature difficult to swallow, providing a physical defense while leaving the rest of its body flexible and mobile. Along the ventral side, Hallucigenia possessed a series of slender, flexible appendages which were initially misunderstood. For decades, paleontologists debated which end was the head and which the tail, and which structures were legs versus spines. Today, it is understood that the creature walked on these delicate limbs, moving cautiously along the sea floor. Its gait was likely slow and deliberate, a necessity in the predator-rich environment of Cambrian oceans, and it may have used its limbs to probe the sediment for small prey, including microbial mats and tiny invertebrates. The head of Hallucigenia is a feature of particular intrigue. Fossil evidence shows a small, simple mouth at one end, capable of ingesting microscopic food. It lacked jaws, teeth, or sophisticated sensory organs, relying instead on a combination of tactile feedback and chemical detection to navigate and forage. Its spines provided passive defense, but without active vision or other advanced senses, Hallucigenia's survival depended on its ability to remain concealed, moving slowly and deliberately along the seafloor. Hallucigenia's discovery illustrates the challenges of interpreting Cambrian life. Early reconstructions famously placed the animal upside down, with its spines imagined as legs and its limbs projecting skyward. For decades, scientific illustration depicted a creature walking on rigid dorsal spikes, a design that seemed almost impossible. Only careful examination of new fossils, coupled with insights from related organisms, clarified its true orientation. The correct reconstruction revealed a small, spiny predator with a delicate but functional body plan, perfectly suited for the Cambrian environment. The ecological role of Hallucigenia provides insight into the dynamics of early animal communities. It occupied the lower tiers of the food web, feeding on microorganisms and detritus, while avoiding predation through its spines and cryptic behavior. Its presence shaped the evolution of both predators and competitors, creating selective pressure for specialized hunting strategies. Larger predators, such as Anomalocaris, may have found Hallucigenia challenging to capture or ingest, demonstrating that even small animals could exert influence on the structure of Cambrian ecosystems. Fossils of Hallucigenia reveal further details about its evolutionary relationships. Once thought to be an isolated anomaly, it is now recognized as part of the lobopod lineage, a group of worm-like animals that represent a transitional stage between soft-bodied invertebrates and the more complex arthropods that would dominate later seas. Its spines foreshadow the defensive adaptations of later organisms, while its walking limbs suggest an early experimentation with locomotion in segmented animals. This combination of traits, both primitive and specialized, makes Hallucigenia a living record of evolution in progress. Soft tissue preservation in the Burgess Shale allows paleontologists to examine minute anatomical details, including the arrangement of muscles, the articulation of limbs, and the placement of spines. These details provide a window into the life of an animal that thrived 500 million years ago. Even subtle differences in spine curvature or limb length may have influenced its survival, suggesting that Hallucigenia's morphology was finely tuned to environmental pressures, predation, and feeding efficiency. The extinction of Hallucigenia remains a subject of speculation. As Cambrian ecosystems became more complex and competition intensified, the unique body plan of Hallucigenia may have limited its adaptive potential. Larger, faster predators and more versatile arthropods could exploit similar resources more efficiently, leaving Hallucigenia confined to narrower ecological niches. Its disappearance underscores the ephemeral nature of evolutionary experiments. Successful for a time, yet vulnerable to shifts in ecosystem dynamics. Despite its brief tenure in the fossil record, Hallucigenia offers profound lessons about the Cambrian world. It demonstrates that life during this period was not constrained by conventional forms, instead exploring possibilities that seem strange or impossible to modern eyes. Its combination of defensive spines, delicate limbs, and simple feeding structures reflects a balance of survival strategies that allowed it to persist in a challenging environment. Fossilized in stone, Hallucigenia continues to captivate scientists and enthusiasts alike, reminding us of a time when the oceans were a canvas for evolution's most daring experiments. In contemplating Hallucigenia, one is struck by the ingenuity of life's solutions to survival. Its body plan, so unlike any seen today, reflects both constraint and creativity, a design shaped by predation, resource availability, and the pressures of a competitive world. While the creature itself vanished, its legacy persists in the insights it provides into early animal evolution, the dynamics of Cambrian ecosystems, and the boundless inventiveness of life itself. Pelecyphorus, the Cambrian's hidden swimmer. The Cambrian seas were a mosaic of life, filled with shallow reefs, tidal channels, and soft sediment plains. Among the early animals navigating these waters was Pelecyphorus, a little-known yet remarkable swimmer whose fossil traces reveal a creature adapted for both stealth and efficiency. Though far smaller than giants like Anomalocaris, its streamlined body, lateral lobes, and delicate appendages made it an accomplished predator in its own right. Exploiting the abundance of microbial mats and soft-bodied prey that carpeted the seafloor. Pelecyphorus's anatomy was a study in subtle specialization. Its elongated body was encased in a flexible, lightly sclerotized covering, with a series of overlapping lateral lobes that functioned as swimming flaps. Each flap undulated in a coordinated wave, producing forward motion while minimizing energy expenditure. Fossil impressions indicate fine ridges along the edges of these lobes, likely increasing surface area for propulsion and allowing for controlled maneuvering. In a crowded seascape, such agility could mean the difference between a successful hunt and becoming prey. The head of Pelecyphorus was dominated by a pair of compound eyes mounted on short, lateral stalks. Unlike the panoramic vision of Opabinia, these eyes were positioned for forward-focused hunting, giving the animal depth perception and the ability to track moving targets. Tiny grasping appendages, located just beneath the eyes, suggest a predatory strategy reliant on quick snatches of prey rather than prolonged pursuit. Fossilized remnants of these limbs show delicate spines, indicating both tactile and grasping function adaptations that allowed Pelecyphorus to extract small organisms from cracks in the sediment. Despite its small size, Pelecyphorus occupied a distinct ecological niche. The Cambrian seas were filled with filter feeders, grazers, and detritivores, yet Pelecyphorus hunted actively, navigating tight spaces and ambushing soft-bodied prey. Its efficiency demonstrates that size alone was not a determinant of dominance in these early ecosystems. Even diminutive predators could influence community structure, driving defensive adaptations in smaller organisms and influencing patterns of resource distribution. Preservation of Pelecyphorus fossils is rare, but the Burgess Shale and Chengjiang deposits offer enough material to reconstruct its lifestyle. Fossil specimens reveal the flexibility of its body, the articulation of its swimming flaps, and the delicate nature of its feeding appendages. Subtle features, such as the curvature of the lobes and the alignment of spines, provide clues to its swimming mechanics, suggesting that the animal could make sudden turns and adjust its depth with precision, a necessity for evading larger predators and successfully capturing prey. The evolutionary relationships of Pelecyphorus remain partially obscured. While clearly related to other lobopodians and anomalocaridid relatives, its combination of features is unique. The animal demonstrates both primitive characteristics, such as a soft, elongated body and simple appendages, and specialized adaptations, such as refined lateral lobes and grasping limbs. This mixture of traits highlights the experimental nature of Cambrian evolution, when body plans were still fluid and organisms explored configurations that would later be abandoned or refined in other lineages. Ecological pressures shaped every aspect of Pelecyphorus's anatomy. Its sensory organs allowed it to detect prey in low-light conditions, while its lateral lobes enabled sustained swimming over the substrate. The subtle mechanics of its body also suggest a form of energy-efficient cruising, allowing it to patrol large areas without exhausting itself. In a period dominated by both predators and prey, experimenting with novel survival strategies, such efficiency would have been essential for persistence. Extinction eventually claimed Pelecyphorus, as it did many other Cambrian creatures. The rise of more specialized predators, environmental shifts, and competition from emerging arthropods gradually reduced the abundance of lobopodians like this species. Yet the fossil record preserves the story of its life and adaptations, offering a window into a time when evolutionary innovation knew few bounds. Through Pelecyphorus, paleontologists gain insight into the subtleties of Cambrian ecosystems. Its combination of stealth, agility, and predation demonstrates that not all survival depended on size or brute force. Instead, refinement of body mechanics, sensory acuity, and habitat specialization were equally effective strategies. In the silent depths of the Cambrian seas, Pelecyphorus moved with efficiency, a testament to the ingenuity and diversity of early life, leaving a legacy that continues to inform our understanding of evolution's earliest experiments. Marella, the spiny ambassador of the Cambrian. The Cambrian seas were a theatre of evolutionary experimentation, where the boundaries of form and function were tested in every niche. Among the most iconic residents of these early oceans was Marella splendens, a small yet astonishingly complex arthropod that exemplifies the ingenuity of early animal life. Measuring only a few centimetres in length, Marella is often called the lace crab by researchers, though its body bears little resemblance to any modern crustacean. Preserved in extraordinary detail in the Burgess Shale, Marella offers scientists a vivid snapshot of how life adapted to the challenges of the Cambrian world. Marella's body was segmented and covered with a chitinous exoskeleton, yet its most striking feature was a series of long, delicate spines protruding from the dorsal side. These spines, arranged symmetrically along the thorax, likely served as a deterrent against predators, giving it a spiky silhouette that would have made swallowing or attacking the animal difficult. Fossils show variations in spine length and curvature, suggesting some degree of flexibility or specialization, possibly linked to defense, display, or stability during swimming. The fine preservation of these spines allows paleontologists to study their microstructure, revealing how evolution crafted both protective and functional elements in a small animal. Alongside its defensive adaptations, Marella possessed multiple pairs of biramous appendages, limbs branching into two distinct structures that enabled both locomotion and feeding. These limbs likely allowed the animal to scuttle across the seafloor or swim through shallow currents with remarkable dexterity. Fossil evidence suggests that some appendages bore delicate setae, functioning like bristles, which may have helped capture microscopic prey or filter food particles from the water. This dual role of limbs, combining movement and feeding, highlights a level of specialization that reveals how even small animals could master multiple ecological strategies simultaneously. The head of Marella is equally remarkable. Large, stalked, compound eyes provided wide visual coverage, while antennae extended forward, equipped with sensory hairs to detect chemical and tactile cues in the surrounding water. Together, these sensory organs allowed Marella to navigate complex environments, avoid predators, and locate food in the dense Cambrian communities of sponges, microbial mats, and soft-bodied animals. Its ability to process multiple sensory inputs simultaneously made it an agile and efficient inhabitant of the shallow seas. Marella's ecological role extended beyond predation and self-defense. Its interactions with the environment influenced the distribution and behavior of other organisms. By grazing on microbial mats, it may have shaped the composition of benthic communities, altering nutrient cycling and sediment structure. Its presence would have been a constant factor in predator-prey dynamics, both as a target for larger hunters and as a competitor for smaller scavengers and detritivores. In this way, Marella acted as a subtle but essential component of its ecosystem, contributing to the complex web of life that defined the Cambrian seas. Fossil specimens provide remarkable insights into Marella's behavior and life history. Some show evidence of molting, a necessary process for growth in arthropods, with remnants of old exoskeletons preserved alongside fully articulated individuals. Others reveal minute details of limb articulation and setae orientation, offering clues to feeding strategies and locomotion. These fossils allow scientists to reconstruct not only the animal's anatomy, but also its likely movements, feeding habits, and responses to environmental pressures. Marella's evolutionary relationships continue to fascinate researchers. While clearly an arthropod, it does not fit neatly into any modern group. Its combination of spines, biramous limbs, and sensory organs represents a mosaic of features, some primitive, some highly specialized. It provides a window into a stage of arthropod evolution when body plans were highly experimental, a time before the dominance of modern crustaceans, insects, and arachnids. Marella demonstrates that complexity and specialization arose far earlier than previously assumed, revealing the ingenuity of early animal life. Despite its small size, Marella's survival strategy was effective. Its combination of defense, mobility, and sensory sophistication allowed it to navigate a world filled with predators like Anomalocaris and larger lobopodians, while exploiting resources that might have been inaccessible to other creatures. Yet, like many Cambrian animals, it eventually disappeared from the fossil record, replaced by more specialized or resilient lineages. Its extinction underscores the transient nature of evolutionary experiments, a reminder that adaptation is not a guarantee of permanence. Morella captivates scientists and enthusiasts alike because it embodies the Cambrian ethos: experimentation, adaptation, and innovation. Its spiny armour, complex limbs, and finely tuned sensory apparatus reveal an animal perfectly adapted to its environment, yet one that does not resemble any modern counterpart. Fossilized in exquisite detail, it serves as both a window into the past and a symbol of evolution's daring creativity, a small but extraordinary ambassador of a time when life first began exploring the astonishing diversity of forms that would shape Earth's oceans for millions of years to come. Leancoilia, the swift predator of the Cambrian. Amid the bustling diversity of Cambrian seas, where every nook and cranny of the seafloor teemed with life, Leancoilia superlata carved out a niche as one of the most agile and enigmatic predators of its time. Measuring roughly 10 centimeters in length, this small but formidable arthropod combined speed, precision, and a uniquely sophisticated body plan that allowed it to thrive in a competitive environment crowded with soft-bodied prey and lurking predators. Fossil evidence from the Burgess Shale and Chengjiang deposits reveals not only the structural complexity of Leancoilia, but also hints at the ecological pressures that shaped its evolution. The body of Leancoilia was segmented, elongated, and covered with a lightly sclerotized exoskeleton, offering a balance of flexibility and protection. Along its sides were pairs of long, paddle-like appendages, highly articulated and tipped with fine bristles. These limbs functioned both as swimming aids and as stabilizers, enabling the animal to maneuver with exceptional agility. In an environment dominated by predators such as Anomalocaris, such maneuverability was not merely advantageous, it was essential. Fossilized imprints show subtle variations in appendage length and orientation, suggesting that Leancoilia could make rapid directional changes while maintaining stability in strong currents, effectively evading attacks while pursuing its own prey. At the front, the head bore a pair of large, compound eyes mounted on short stalks. These eyes were finely tuned for detecting movement in the water column and across the sea floor. Their placement provided a wide field of vision, allowing Leancoilia to track prey or spot approaching predators with minimal delay. Between the eyes extended a pair of grasping appendages, armed with spines and articulated joints capable of delicate, precise movements. Fossil evidence suggests that these limbs were highly versatile, able to reach into crevices or scoop small animals from sediment, combining speed and finesse in a manner unmatched by many of its contemporaries. The trunk of Leancoilia contained multiple pairs of biramous appendages, specialized for locomotion and food capture. These limbs likely helped it hover above the seafloor, sweep the substrate for microbial mats, and extract tiny prey from hidden crevices. The combination of dorsal and lateral flaps with jointed legs indicates a sophisticated control over movement in 3 dimensions, a rare capability for Cambrian animals. In life, Leancoilia would have appeared as a fluid, almost graceful predator, moving with calculated precision through its crowded ecosystem. One of the most striking features of Leancoilia is its tail structure. Narrow and flexible, it likely provided fine adjustments to balance and orientation while swimming. Fossil specimens preserved in lateral view reveal subtle muscle impressions and the articulation of the tail segments, suggesting that Leancoilia could make rapid twists and turns, an essential adaptation for both hunting and evading predators. Its entire body seems optimized for efficiency, combining speed, sensory awareness, and precise limb coordination in an animal small enough to exploit ecological niches inaccessible to larger predators. Leancoilia's ecological role illustrates the intricate dynamics of Cambrian communities. While it hunted smaller, soft-bodied organisms, it was itself prey for larger predators. Its presence influenced the behavior of other animals. Small grazers might have taken refuge under microbial mats, while other arthropods adapted their locomotion to avoid detection. In this web of interactions, Liancolia was both predator and prey, a vital component in the shaping of early marine ecosystems. Its adaptations demonstrate that survival in the Cambrian seas required a delicate balance of speed, awareness, and specialized morphology. Fossil evidence continues to reveal the subtleties of Liancoilia's lifestyle. Some specimens show partial exoskeletons, indicating molting behavior essential for growth, while others preserve the delicate bristles and spines of appendages in fine detail. Analysis of limb orientation and body segmentation suggests coordinated movement patterns, likely used both for navigation and hunting. These insights allow paleontologists to reconstruct not just anatomy, but behavior, providing a rare glimpse into the daily life of a predator that thrived over 500 million years ago. The evolutionary position of Liancoilia underscores the experimental nature of Cambrian evolution. It shares characteristics with early arthropods, but also exhibits unique features such as highly specialized grasping appendages and finely tuned swimming flaps that distinguish it from related species. These traits highlight the creative experimentation occurring in early animal lineages, where natural selection explored solutions that were simultaneously effective and ephemeral. Liancoilia's lineage would eventually vanish, leaving behind no direct descendants, yet its fossil record preserves a testament to the ingenuity of early arthropod design. By studying Liancoilia, scientists gain insight into the principles that governed Cambrian ecosystems: specialization, adaptability, and precision in morphology and behavior. Its combination of sensory acuity, agile locomotion, and specialized appendages allowed it to thrive in a crowded seafloor filled with both opportunity and danger. Fossilized in the Burgess Shale, it stands as a vivid example of the delicate balance of form and function that evolution can achieve. A small predator whose life illuminates the complexity and inventiveness of the Cambrian explosion. Liancoilia reminds us that evolutionary success in early seas required more than size or brute strength. Efficiency, adaptability, and the ability to exploit subtle ecological niches were equally critical. In the dance of predator and prey, this small arthropod moved with precision, leaving behind a legacy of innovation and complexity that continues to inform our understanding of life's early experiments. Wiwaxia, the armoured grazer of the Cambrian, amid the undulating microbial mats and soft sediment plains of the Cambrian seafloor, a creature moved with quiet efficiency, leaving behind rows of impressions that would captivate scientists for decades. Wiwaxia corrugata, a small yet extraordinary organism, combined a protective armour of dorsal scales with delicate spines and an unusual feeding apparatus, making it one of the most intriguing grazers of its time. Measuring just a few centimetres in length, Wiwaxia appears modest in size, yet its body reveals a level of structural sophistication that challenges assumptions about early animal complexity. The most striking feature of Wiwaxia is its armour. Fossil evidence shows a series of overlapping sclerites, scale-like structures arranged along the dorsal surface, interspersed with long, rigid spines. These spines, extending outward in pairs, provided defence against predators and may have served as deterrents against attacks from above. Some fossils suggest variations in spine length across individuals, hinting at potential sexual dimorphism or ontogenetic changes as the animal grew. The dorsal armour, lightweight yet resilient, allowed Wiwaxia to remain agile while navigating complex terrain, illustrating a balance between protection and mobility that would characterize many later lineages of armored animals. Beneath its armor, Wiwaxia possessed a soft, segmented body capable of undulating movements. Along its ventral surface were numerous simple, leg-like structures, likely used to crawl across the seafloor and maintain contact with the sediment while grazing. These limbs, in combination with body flexion, allowed it to move efficiently over microbial mats, detritus, and fine sediments, while maintaining stability against water currents and predators. Fossils show impressions of the ventral surface and limb positions, providing paleontologists with detailed insights into its locomotion and behavior. The feeding apparatus of Wiwaxia was equally remarkable. Near the anterior end, it bore a radula-like structure, a toothed, rasping organ used to scrape microbial layers from the substrate. This apparatus suggests that Wiwaxia was a specialized grazer, feeding primarily on microscopic algae, bacteria, and detritus that coated the Cambrian seafloor. The combination of armored protection and efficient grazing allowed Wiwaxia to exploit a niche that few other animals could occupy, shaping both its survival strategy and the composition of benthic communities. Wiwaxia's ecological role was subtle but significant. By grazing on microbial mats, it influenced the structure and distribution of these communities, potentially affecting nutrient cycling and sediment composition. Its armored body made it less vulnerable to predators such as Anomalocaris and other larger lobopodians, allowing it to persist where unprotected grazers might have been eliminated. Fossilized trackways suggest deliberate movements across the sea floor, providing a window into its interactions with the environment and the strategies it employed to survive in a dynamic ecosystem. The evolutionary significance of Wiwaxia remains a subject of ongoing study. Its combination of dorsal armor, spines, and grazing apparatus places it among the stem group mollusks or lobopod relatives, though its exact placement is still debated. The animal demonstrates early experimentation with body plans that combine protection, mobility, and specialized feeding, highlighting the creative exploration of evolutionary solutions during the Cambrian explosion. Fossil evidence allows scientists to trace the origins of key morphological innovations that would later influence mollusks, annelids, and other animal groups. Fossil preservation is key to understanding Wiwaxia's biology. Many specimens retain the delicate arrangement of sclerites and spines, as well as impressions of the soft body beneath. These fossils show that the armor was not rigidly fixed, but lightly articulated to allow body flexion, enabling locomotion without compromising protection. Minute details of spine microstructure and radular morphology reveal functional adaptations, providing insight into feeding mechanics, predator avoidance, and ecological interactions. Wiwaxia's extinction, like that of many Cambrian organisms, illustrates the transient nature of evolutionary experiments. As ecosystems became more complex, with increasing competition and the emergence of more efficient grazers and predators, Wiwaxia's niche may have been outcompeted. Yet its fossil record preserves an elegant solution to a persistent problem: how to feed safely and efficiently in a predator-rich environment. Its legacy endures not only in the fossils themselves, but also in the evolutionary lessons they provide, illustrating the early experimentation with armor, mobility, and feeding specialization. Through the study of Wiwaxia, scientists gain a profound appreciation for the ingenuity of early life. Its combination of protective armor, specialized feeding apparatus, and subtle locomotor adaptations demonstrates a level of sophistication unexpected for such small organisms. In the quiet sediments of the Cambrian seafloor, Wiwaxia grazed and moved with precision, contributing to the balance and structure of its ecosystem, a small but remarkable participant in the first great explosion of complex animal life. Even after more than 500 million years, Wiwaxia continues to captivate paleontologists and enthusiasts alike. Its armoured body, feeding specialization, and ecological role provide a vivid portrait of life, experimenting with form and function. Fossils frozen in stone preserve not only its anatomy, but also its story. A tale of survival, adaptation, and evolutionary creativity in a world still discovering the possibilities of complex life. Odaraya, the sprinting swimmer of the Cambrian. The Cambrian seas were alive with experimentation, a dynamic world where form and function were constantly being tested. Among the most remarkable of these early marine innovators was Odaraya alata, a small but highly specialized predator whose long, streamlined body and powerful swimming appendages allowed it to navigate the dense, predator-filled waters with extraordinary agility. Measuring roughly 10 centimeters in length, Odaraya may seem modest at first glance, yet its anatomy reveals a creature perfectly adapted to life in a highly competitive ecosystem, combining speed, precision, and evolutionary ingenuity. Odaraya's body was elongated, segmented, and laterally compressed, creating a hydrodynamic profile that minimized drag as it moved through the water. Along each side, pairs of wide, flattened flaps acted as swimming limbs, moving in coordinated undulations that propelled the animal forward with surprising speed. These flaps were not only powerful but highly maneuverable, allowing Odaraya to make rapid turns, sudden ascents, or swift dives in pursuit of prey or in evasion of larger predators such as Anomalocaris. Fossil evidence reveals subtle variations in flap size and curvature, indicating precise adaptation to its environment and highly efficient locomotion mechanics. The head of Odaraya was dominated by a pair of stalked, compound eyes, positioned to provide both forward and lateral vision. These eyes contain numerous lenses, giving the animal a broad field of view and the ability to detect even subtle movements in the water column. Such advanced sensory capacity suggests that Odaraya was an active predator, relying on sight to track prey and avoid threats. Just beneath the eyes, delicate anterior appendages functioned as grasping tools, capable of seizing small arthropods, larvae, or other soft-bodied organisms. The combination of high-speed swimming and precision appendages allowed Odaraya to hunt efficiently while minimizing energy expenditure, a crucial advantage in the resource-competitive Cambrian seas. Fossilized specimens of Odaraya provide fascinating insights into its ecology. Many individuals are preserved in lateral view, revealing the arrangement of flaps, the segmentation of the body, and the delicate articulation of the anterior appendages. These details suggest that Odaraya could maintain stability during rapid swimming and execute fine-scale adjustments while capturing prey. Its morphology indicates a life spent almost entirely in the water column, hovering above the sea floor to ambush prey or avoid benthic predators, rather than crawling along sediments like many of its contemporaries. Odaraya's ecological niche was unique. Its combination of speed, sensory awareness, and grasping ability allowed it to exploit small prey that were inaccessible to slower or less maneuverable animals. By preying on larvae, small arthropods, and soft-bodied organisms, it shaped the structure of Cambrian food webs, influencing the distribution and behavior of multiple species. Predators and prey alike had to navigate the threat posed by Odaraya's swift, precise hunting, making it a key player in the balance of early marine ecosystems. The evolutionary significance of Odaraya is particularly compelling, While clearly related to other early arthropods, its adaptations from highly efficient swimming flaps to stalked compound eyes reflect an advanced level of specialization. It represents an early experiment in active mid-water predation, predating more sophisticated swimming arthropods by millions of years. Its body plan illustrates the flexibility and innovation of early animal evolution, where even small creatures could develop specialized strategies to dominate their niches. Preservation in the Burgess Shale allows paleontologists to study minute details of Odoria's anatomy. Impressions of soft tissues, flap articulation, and anterior appendages reveal the mechanics of swimming and prey capture. Analysis of these fossils suggests that Odaria combined energy-efficient cruising with sudden bursts of speed, enabling both ambush and pursuit predation. This dual strategy illustrates a level of behavioral sophistication rare in animals of this early period and highlights the selective pressures driving innovation during the Cambrian explosion. Odoria's extinction underscores the transient nature of evolutionary success. As ecosystems became more complex and competition intensified, many specialized predators faced new challenges. Larger, more versatile arthropods and evolving lobopods likely encroached on Odoria's ecological niche, eventually reducing its population and driving it to extinction. Yet the fossil record preserves a striking example of evolutionary experimentation. A small, agile predator that combined speed, sensory acuity, and precise hunting adaptations in a way that would not be replicated for millions of years. Studying Odoria provides profound insights into early marine ecosystems. Its anatomy and behavior demonstrate that success in Cambrian seas required more than size or brute strength. Precision, agility, and the ability to exploit specific ecological niches were equally critical. Fossils frozen in time allow scientists to reconstruct not only its physical form, but also its ecological interactions, revealing a predator that was both highly specialized and remarkably effective. Odoria reminds us that evolutionary innovation was not uniform but experimental. Each creature of the Cambrian seas was exploring its own solution to survival, and Odoria represents one of the most successful strategies of mid-water predation. Its streamlined body, powerful swimming flaps, and keen vision illustrate a creature finely tuned to its environment, thriving in a world of constant danger and opportunity. In the currents of the ancient seas, Odoria moved with grace and efficiency, leaving behind a fossil record that continues to inspire and challenge our understanding of early animal evolution. Herdia, the titanic predator of the Cambrian. The Cambrian oceans were a theatre of evolutionary experimentation, where speed, sensory adaptation, and predatory innovation determined survival. Among the giants of this era was Herdia victoria, a formidable predator that combined massive size, powerful appendages, and a complex body plan to dominate its ecological niche. Unlike the smaller, more agile arthropods that populated the seas, Herdia could reach lengths of over 1/2 meter, making it one of the largest predators of the Cambrian. Fossils from the Burgess Shale and related deposits offer a window into its anatomy and lifestyle, revealing a predator perfectly adapted to the challenges of an early marine ecosystem. Herdia's most striking feature is its frontal carapace, a large, crescent-shaped shield that extended from the head region. This structure housed the mouth and feeding apparatus, forming an efficient trap for capturing prey. Along the front edge, paired frontal appendages, jointed and spined, could sweep outward, guiding small arthropods, worms, and soft-bodied animals into the mouth. The morphology of these appendages suggests both power and precision. capable of grasping, holding, and manipulating prey with remarkable dexterity. Fossil evidence indicates a curvature in the spines that would have prevented prey from escaping, creating a predatory efficiency unparalleled in its time. Beneath the frontal appendages, Herdia's mouth was circular, lined with overlapping plates that functioned like crushing teeth. This oral cone allowed the predator to consume organisms with light armor or exoskeletons, adding versatility to its diet. Unlike the simplistic mouths of smaller Cambrian predators, Herdia's feeding system combined grasping, guidance, and crushing into a coordinated mechanism, making it capable of handling a variety of prey types. The fossil record preserves subtle impressions of these structures, giving paleontologists detailed insight into how this predator interacted with its environment. The body of Herdia was long and segmented, with lateral flaps running the length of the trunk. These flaps, each bearing delicate, gill-like structures, enabled swimming through coordinated undulations, providing both propulsion and respiration. The flaps' arrangement and flexibility suggest that Herdia could hover over the seafloor, glide through mid-water, or execute bursts of speed to ambush prey. Unlike smaller predators that relied on quick maneuvers alone, Herdia combined size, strength, and controlled locomotion, making it a predator capable of dominating multiple ecological zones. Eyes of Herdia were stalked and compound, positioned to provide an expansive field of vision. These organs enabled the predator to detect movement across a wide range, crucial for tracking prey in dimly lit Cambrian waters. Paired with the frontal appendages, Herdia's sensory system created a sophisticated feedback loop: detect, target, and capture. Its eyes reveal that even the earliest large predators possessed complex visual systems, suggesting that sensory innovation was critical in early marine ecosystems. Herdia's ecological role extended beyond predation. By preying on smaller arthropods and soft-bodied organisms, it influenced the distribution, behavior, and evolution of numerous species. Fossil evidence of contemporaneous prey shows defensive adaptations such as spines, burrowing behavior, and evasive swimming strategies, suggesting a direct evolutionary arms race between Herdia and its targets. This dynamic underscores the Cambrian seas as not merely a stage for individual survival, but a crucible of rapid evolutionary experimentation, where selective pressures drove morphological innovation on multiple fronts. Fossils preserve further details about Herdia's locomotion. Impressions of the lateral flaps indicate fine articulation, allowing subtle control over direction and speed. The tail region, tapering yet flexible, likely provided stability and facilitated rapid changes in pitch and yaw during swimming. Such adaptations reveal that Herdia was not a clumsy brute, but a highly efficient predator, capable of coordinating body movements, sensory input, and feeding actions simultaneously. Its size, paired with agility, made it a dominant force in Cambrian seas. The evolutionary significance of Herdia is profound. As a member of the Anomalocaridid family, it represents one of the earliest examples of large, active predators. Its combination of grasping appendages, specialized oral cone, and flexible swimming flaps illustrates a level of specialization unmatched by most contemporaries. Herdia demonstrates that early arthropod evolution was not simply incremental, it was experimental, with lineages exploring forms and functions that would not be replicated for hundreds of millions of years. Extinction eventually claimed Herdia as ecosystems shifted and competition intensified. The rise of more efficient arthropods and changing environmental conditions likely reduced its population, leaving no direct descendants. Yet the fossil record preserves a creature of remarkable design, a predator that combined size, sensory sophistication, and mechanical precision in a way that anticipated the complexity of later marine predators. Herdia reminds us that the Cambrian explosion was not solely about small creatures or simple body plans. It was a period when evolution dared to experiment with scale, function, and predation. Its fossils, frozen in time, reveal the mechanics of a predator perfectly adapted to its world, illustrating the interplay of anatomy, behavior, and ecological influence. In the deep currents of Cambrian seas, Herdia moved with deliberate power, a titanic predator whose life illuminates the ingenuity, complexity, and daring of early animal evolution. Amplectobelua, the grasping giant of the Cambrian. The Cambrian seas were a crucible of innovation, a world where life experimented with form, function, and survival in ways both subtle and extraordinary. Among the apex predators navigating these ancient waters was Amplectobelua cymbrachiata, a close relative of the anomalocaridids, renowned for its formidable grasping appendages, elongated segmented body, and extraordinary hunting capabilities. Fossils from the Burgess Shale and other deposits reveal a creature finely tuned for predation, combining speed, precision, and sensory acuity in a predator that dominated its ecological niche. Amplectobelua's most striking adaptation was its pair of robust, spined frontal appendages. These specialized limbs, long and jointed, functioned like mechanical graspers, capable of seizing, holding, and manipulating prey with deadly precision. The appendages were equipped with inward-facing spines that prevented captured organisms from escaping, while joint articulation allowed complex movement patterns, enabling the predator to strike with both speed and accuracy. Fossilized specimens show wear patterns and curvature that hint at repeated use in hunting, suggesting these appendages were the primary tools for securing a meal. The oral cone of Amplectobelua was circular and reinforced with plate-like structures, designed to crush or process soft-bodied prey. Unlike smaller predators with simple mouths, Amplectobelua could handle a wider range of prey, from armored arthropods to delicate worms, showcasing versatility that made it one of the top predators of its ecosystem. Fossil evidence preserves subtle impressions of the oral plates, revealing complex arrangements that maximized the predator's efficiency. The combination of grasping appendages and a powerful oral cone made Amplectobelua a formidable hunter, capable of exploiting multiple prey types simultaneously. Its elongated, segmented body was streamlined for swimming, with pairs of lateral flaps running the length of the trunk. These flaps provided propulsion, maneuverability, and stability, allowing Amplectobelua to hover above the seafloor, chase prey through the water column, or make sudden, evasive maneuvers to avoid larger predators. Impressions of gill-like structures beneath the flaps suggest that locomotion and respiration were integrated, allowing continuous movement without sacrificing oxygen intake, a critical adaptation for an active predator in oxygen-variable Cambrian seas. The eyes of Amplectobelua were large, stalked, and compound, positioned to provide panoramic vision. These organs enabled detection of prey across a wide field, while their size and structure suggest remarkable visual acuity for the period. Paired with its grasping appendages, the predator could execute coordinated hunting strategies, detecting prey from a distance, closing in rapidly, and capturing it with precision. Fossils indicate eye placement optimized both forward and lateral vision, giving Amplectobelua a critical advantage in the crowded, competitive Cambrian marine environment. Ecologically, Amplectobelua played a key role as both predator and evolutionary driver. By preying on diverse small animals, it influenced the distribution, behavior, and evolution of multiple species. Prey animals evolved protective spines, burrowing behaviors, and evasive locomotion in response to predators like Amplectobelua. This arms race illustrates the dynamic nature of Cambrian ecosystems, where predation pressures drove rapid experimentation and adaptation. Fossilized trackways and gut contents provide insight into diet and hunting strategies, confirming that Amplectobelua was both an opportunistic feeder and a specialized predator. Fossil evidence further reveals details about Amplectobelua's locomotion and feeding mechanics. The lateral flaps were articulated for controlled undulations, and the tail region provided steering and stabilization. Together, these features allowed the predator to hover, sprint forward, or make agile turns, coordinating body movements with the strike of its frontal appendages. Minute structures, including bristles on appendages and ridges on the oral cone, illustrate finely tuned adaptations for both prey capture and environmental navigation. The evolutionary significance of Amplectobelua cannot be overstated. Its combination of grasping appendages, reinforced oral cone, and efficient locomotor system represents an advanced predatory strategy rarely seen in Cambrian animals. As a member of the anomalocaridid lineage, it highlights the remarkable experimentation occurring in early arthropod evolution, blending primitive features with highly specialized adaptations. The creature illustrates how early predators could combine size, agility, and precision in ways that influence the structure of entire ecosystems. Extinction eventually claimed Amplectobelua as ecosystems evolved and competition intensified. The rise of more versatile predators, environmental changes, and shifting prey availability likely contributed to its decline. Yet the fossil record preserves a vivid portrait of a predator whose adaptations reflect both evolutionary creativity and ecological dominance. Amplectobelua remains a testament to the ingenuity of Cambrian life, embodying the delicate balance between form, function, and survival in one of Earth's earliest marine ecosystems. Through the study of Amplectobelua, scientists gain insight into the interplay between anatomy, behavior, and environment. Its grasping appendages, panoramic vision, and streamlined body reveal a predator capable of precision hunting, agile maneuvering, and ecosystem-wide influence. Fossilized in exquisite detail, it offers a window into the complexity of Cambrian predation, demonstrating that early life was not merely experimenting with form, but also with strategy, efficiency, and ecological mastery. Amplectobelua's story reminds us that evolutionary success during the Cambrian explosion depended on innovation at multiple levels: sensory acuity, mechanical specialization, and behavioral adaptation. In the shadowy waters of the Cambrian seas, this predator moved with deadly efficiency, shaping both prey and predator lineages alike, leaving a legacy etched in stone, a testament to the remarkable creativity of early life on Earth. Thomisiocharys, the filter-feeding marvel of the Cambrian. In the shadowy Cambrian seas, while swift predators like Amplectobelua dominated mid-water hunting, another lineage of anomalocaridids was experimenting with a radically different feeding strategy. Thomisiocharys borealis, a filter-feeding giant, evolved specialized adaptations to capture microscopic prey suspended in the water column. Unlike its hunting relatives, Thomisiocharys relied not on speed and grasping appendages, but on intricate filtration structures that allowed it to sweep vast volumes of water in search of plankton and small organisms. Its fossilized remains, preserved in the Burgess Shale and Sirius Passet deposits, offer a glimpse into a highly specialized life strategy that transformed early marine ecosystems. Thomisiocharys could grow to lengths of over 0.5 meter, yet its body was designed more for efficiency than brute power. Its elongated, segmented trunk was equipped with a series of lateral flaps that undulated in a coordinated rhythm, propelling the animal through the water with minimal energy expenditure. These flaps, paired with gill-like structures, allowed the animal to maintain both locomotion and respiration simultaneously, an essential adaptation for a creature that relied on continuous movement to feed. Fossils reveal fine details of flap articulation, indicating subtle control over depth, direction, and stability as the animal swept through Cambrian currents. The most remarkable adaptation of Thomisiocharys was its feeding apparatus. Its frontal appendages, unlike the grasping limbs of its predatory cousins, were elongated and lined with closely spaced spines, forming a comb-like structure. These appendages acted as sieves, filtering small organisms such as plankton, tiny arthropods, and other suspended particles from the water. By sweeping these appendages rhythmically, Thomisiocharys could capture significant quantities of prey without the need for pursuit or struggle. Fossil evidence shows slight variations in spine spacing, suggesting adaptations to different prey sizes or water conditions, reflecting a highly specialized and efficient feeding strategy. The head of Thomisiocharys was crowned with large, stalked, compound eyes that provided panoramic vision. While it did not rely on high-speed hunting, these eyes were critical for detecting environmental cues, navigating turbulent waters, and avoiding predators. The combination of visual acuity and precise, filter-like appendages allowed Thomisiocharys to move confidently through mid-water zones, exploiting a resource, suspended microorganisms, that many other animals could not. Fossils indicate a degree of flexibility in the head and appendages, allowing subtle adjustments in feeding angle and orientation to maximize efficiency. Ecologically, Thomisiocharys occupied a unique niche. In contrast to active hunters, it was a passive predator, shaping food webs by consuming abundant planktonic organisms. Its presence influenced population dynamics, providing selective pressure on microorganisms and small arthropods. At the same time, its size and mobility meant it was not immune to predation. Larger anomalocaridids and other apex predators likely viewed Thomisiocharys as a viable target. Fossilized trackways and posture evidence suggest a combination of slow, deliberate movement with occasional bursts of acceleration to escape threats, illustrating a balance between feeding efficiency and survival strategy. The evolutionary significance of Thomisiocharys is profound. It represents one of the earliest known filter-feeding animals, predating similar strategies seen in later marine invertebrates by millions of years. By exploiting a previously untapped resource microscopic plankton, it demonstrates the diversity of ecological experimentation during the Cambrian explosion. Its adaptations highlight a remarkable evolutionary innovation: the transformation of a predatory lineage into a filter-feeding specialist, expanding our understanding of early ecological complexity. Fossil preservation provides remarkable insight into the mechanics of Thomisiocharys. Specimens reveal detailed impressions of the lateral flaps, frontal appendages, and sensory structures, allowing reconstruction of movement patterns and feeding behavior. Analysis of spine arrangement and limb articulation shows how water was channeled efficiently through the appendages, maximizing prey capture while minimizing energy expenditure. These subtle structural features reflect a level of functional sophistication unusual for Cambrian organisms, indicating that evolution had already begun exploring highly specialized ecological strategies. Despite its efficiency, Thomisiocharys, like many Cambrian animals, eventually disappeared from the fossil record. Environmental shifts, increasing competition, and the rise of new predators and filter-feeding organisms likely contributed to its decline. Yet its legacy endures, a testament to the flexibility of early animal life and the boundless creativity of evolutionary experimentation. Thomisiocharys illustrates that survival need not rely solely on speed or predatory aggression. Specialization, efficiency, and exploitation of abundant but overlooked resources were equally powerful strategies. Through the study of Thomisiocharys, scientists gain a window into the delicate balance of Cambrian ecosystems. Its unique adaptations highlight the interplay between morphology, behavior, and environment, demonstrating that early life was capable of incredible ingenuity. Fossils preserve not only its physical form but also its ecological role, revealing a filter-feeding marvel that contributed to the complexity and diversity of the first great explosion of animal life. Thomisiocharys reminds us that evolution is both opportunistic and experimental. By diverging from the predatory strategies of its relatives, it carved a niche that allowed it to thrive in a dynamic and competitive world. In the flowing currents of the Cambrian seas, it swept its comb-like appendages with rhythmic precision, capturing prey invisible to the naked eye and leaving behind a fossil record that continues to inspire awe and curiosity. Stanleycharis, the precision predator of the Cambrian. In the restless Cambrian seas, where evolutionary experimentation reached its peak, few creatures exemplify specialization as elegantly as Stanleycharis hypex. Though modest in size, rarely exceeding 10 centimeters, Stanleycharis was a highly efficient predator, combining precise grasping appendages, panoramic vision, and a streamlined body to dominate its ecological niche. Fossil evidence from the Burgess Shale provides a rare window into its anatomy, feeding behavior, and ecological interactions, revealing a predator finely tuned to a world defined by both opportunity and danger. Stanleycharis's most striking feature was its pair of frontal appendages, robust and spined, capable of seizing prey with extraordinary accuracy. Unlike larger anomalocaridids, whose size alone often determined predatory success, Stanleycharis relied on speed, precision, and coordination. The appendages were jointed and tipped with inward curving spines, allowing them to grasp and hold small arthropods, worms, and soft-bodied animals without crushing them. Fossilized specimens reveal subtle differences in spine morphology among individuals, suggesting adaptive variation that may have corresponded to prey type or developmental stage. These grasping limbs allowed Stanleycharis to hunt with a level of dexterity unusual for Cambrian predators. Its oral cone, circular and reinforced with overlapping plates, allowed it to crush or manipulate prey once secured. Unlike simpler mouths, Stanleycharis's oral cone was capable of processing a range of organisms, from soft-bodied worms to lightly armoured arthropods. Fossil evidence indicates wear patterns on the plates, providing direct insight into feeding mechanics. This combination of grasping appendages and a specialized oral cone illustrates an early integration of precision hunting and mechanical processing, a strategy that enabled Stanleycharis to exploit a diverse array of prey while remaining small and agile. The body of Stanleycharis was elongated and laterally compressed, optimized for swimming. Pairs of lateral flaps extended along the trunk, providing propulsion through coordinated undulations. These flaps were flexible yet powerful, allowing precise adjustments to position, speed, and depth in the water column. Unlike larger, slower predators, Stanleycharis could navigate complex seafloor topography and chase nimble prey through narrow crevices and around obstacles. Fossil evidence shows fine articulation in the flaps, reflecting sophisticated locomotor control that complemented its grasping appendages. Stanleycaris's sensory adaptations were equally remarkable. Its stalked, compound eyes provided near-panorama vision, allowing simultaneous monitoring of predators, prey, and environmental cues. These eyes, unusually large relative to body size, indicate a reliance on acute visual perception in hunting. Fossil analysis suggests that the eyes were highly mobile on their stalks, giving Stanleycaris the ability to track movement without repositioning its entire body to a critical advantage in an environment filled with agile prey and lurking threats. Ecologically, Stanleycaris occupied a niche that required balance between active predation and vigilance. Fossils suggest that it preyed on smaller, soft-bodied organisms, shaping prey distribution and behavior in the surrounding environment. By exerting selective pressure on prey, it influenced the evolution of escape behaviors, defensive spines, and burrowing strategies. Its presence demonstrates the cascading effects a single predator could have on a Cambrian community, underscoring the interconnectedness and complexity of early marine ecosystems. The evolutionary significance of Stanleycaris lies in its integration of multiple specialized traits. Its grasping appendages, articulated oral cone, flexible swimming flaps, and advanced sensory organs collectively illustrate an organism optimized for precision predation. Unlike the large, brute force hunters of its time, Stanleycaris demonstrates that evolutionary success could be achieved through dexterity, efficiency, and subtlety, providing a counterpoint to the size-dominated predatory strategies seen in larger anomalocaridids. Fossil preservation is key to understanding Stanleycaris's lifestyle. Specimens retain detailed impressions of appendage spines, flap articulation, and body segmentation, allowing paleontologists to reconstruct not just anatomy, but probable hunting behavior. Analysis of appendage orientation and eye placement suggests coordinated motion, where grasping, biting, and swimming were finely synchronized. This level of integration implies advanced neural control and an ecological role that was both dynamic and influential. Stanleycaris eventually vanished from the fossil record, its lineage disappearing as ecosystems evolved and competition intensified. Yet it leaves behind a remarkable testament to evolutionary innovation, a predator that combined precision, speed, and sensory sophistication in a small, agile body. Its fossilized remains illuminate the balance between form and function, predator and prey, and adaptation and extinction. Through the study of Stanleycaris, scientists gain insight into the diversity of predatory strategies during the Cambrian explosion. Its specialized morphology and hunting behavior reveal that early ecosystems were shaped not only by size and brute strength, but also by finesse, coordination, and innovation. Fossils provide a vivid record of an organism that moved, hunted, and survived in a complex world, leaving behind a story of evolutionary experimentation frozen in stone. Stanleycaris serves as a reminder that early animal evolution was as much about precision and strategy as it was about scale. Its grasping appendages, panoramic vision, and streamlined body exemplify a creature finely attuned to its environment, navigating the turbulent Cambrian seas with agility and skill. In the currents of ancient oceans, Stanleycaris thrived, hunted, and shaped its ecosystem, an enduring emblem of the ingenuity and diversity of early life. Pamdalurian, the lurking giant of the Cambrian, in the murky, shallow waters of the Cambrian seas, among the reef structures and microbial mats, lived a predator whose subtlety was as striking as its size. Pamdalurian whittingtoni was a large lobopodian, reaching lengths of nearly half a meter, combining a soft, elongated body with lateral flaps and specialized appendages that made it an efficient ambush predator. Fossil evidence from the Burgess Shale provides insight into its anatomy and behavior, revealing a creature that was part predator, part scavenger, navigating a complex ecosystem in ways that continue to challenge modern understanding of early animal life. Pamdalurian's body was elongated, soft, and segmented, with pairs of lateral flaps along the trunk. These flaps, flexible and gill-bearing, provided both locomotion and respiration, allowing the animal to hover just above the sea floor or make subtle undulations to reposition itself for an ambush. Unlike the agile, fast-swimming Anomalocaridids, Pamdalurian relied on stealth and patience, using its moderate swimming ability to adjust position rather than chase prey over long distances. Fossil impressions show detailed flap structure, suggesting a capacity for precise movement and environmental sensing, key for a predator that waited for opportunity rather than forcing it. Its head was adorned with a simple, soft mouth and paired grasping appendages that allowed it to seize soft-bodied prey or scavenge detritus. Unlike the highly specialized frontal appendages of Stanleycaris or Amplectobelua, Pamdalurian's limbs were generalized yet functional, illustrating an evolutionary approach that balanced versatility and predation. Fossils indicate slight differentiation among appendages along the trunk, suggesting that the anterior limbs were optimized for prey capture, while posterior ones aided locomotion and stability. This configuration allowed the animal to coordinate movement and feeding in a way that minimized energy expenditure while maximizing ecological efficiency. Sensory adaptations of Pamdalurian were subtle but effective. While it lacked the stalked compound eyes of many Anomalocaridids, it likely relied on light-sensitive patches or simple visual structures to detect movement, shadows, or changes in the environment. This, combined with mechanosensory input from flaps and body surface, enabled it to detect prey or approaching predators, maintaining a cautious yet opportunistic lifestyle. Fossils preserve impressions that hint at the sensitivity of its flaps and body surface, suggesting an organism finely tuned to its surroundings despite its apparent simplicity. Ecologically, Pamdalurian filled a unique niche. It was neither a pursuit predator nor a filter feeder, but a generalist ambush predator and scavenger, exploiting both live prey and organic material. This dual strategy allowed it to adapt to changing resource availability, moving subtly across the seafloor to exploit opportunities as they arose. Fossil evidence of gut contents indicates consumption of worms and small arthropods, while its morphology suggests scavenging capability, highlighting a versatile lifestyle that allowed it to thrive in diverse Cambrian environments. Evolutionarily, Pamdalurian represents a critical link between early Lobopodians and more derived panarthropods. Its combination of lateral flaps, segmented soft body, and grasping appendages demonstrates an intermediate body plan that blends primitive and specialized traits. This mosaic morphology reveals how early evolution experimented with functional integration, testing solutions in locomotion, respiration, and predation that would influence subsequent lineages. Fossils illustrate subtle articulations and body flexibility, emphasizing the interplay between structure, function, and ecological role in early animal design. Fossil preservation provides unique insights into locomotion and behavior. Impressions show that Pamdalurian could maintain a low profile over sediments, adjusting flaps and body segments to remain concealed while surveying for prey or avoiding predators. Its ambush strategy relied on patience and precise positioning, contrasting sharply with the high-speed predation of its anomalocaridid cousins. Fossilized trackways hint at deliberate, controlled movement across seafloor surfaces, providing a glimpse into the subtle mechanics of Cambrian predation. Despite its advantages, Pamdalurian eventually disappeared from the fossil record. Shifts in predator-prey dynamics, competition from faster, more specialized hunters, and environmental changes likely contributed to its decline. Yet its legacy endures, offering insight into a mode of life that balanced versatility, stealth, and predatory efficiency. It serves as a reminder that evolutionary success need not rely solely on aggression or speed. Subtlety, adaptability, and opportunism could be equally effective strategies in early ecosystems. Through Pamdalurian, scientists gain an appreciation for the diversity of early predatory strategies. Its morphology and behavior illustrate the evolutionary experimentation that characterized the Cambrian explosion, revealing creatures that combined aspects of ambush, scavenging, and active predation in innovative ways. Fossils preserve not only physical structure, but also ecological context, allowing reconstructions of behavior, hunting strategy, and environmental interactions. Pamdalurian exemplifies the ingenuity of early animal evolution. Its combination of lateral flaps, grasping appendages, and ambush strategy demonstrates that success in Cambrian seas required flexibility, precision, and ecological awareness. In the shadowy currents above microbial mats and reef structures, Pamdalurian moved with patient intent, seizing prey, scavenging detritus, and navigating a world alive with predators and competitors. Its fossilized remains endure as a testament to the subtle brilliance of early evolutionary experimentation, a predator that thrived not through raw power, but through versatility, patience, and the quiet mastery of its environment. Kerygmachela, the elusive lobopodian of the Cambrian, in the shallow, sun-dappled waters of the Cambrian seas, a creature moved with quiet precision, navigating reefs and microbial mats with a combination of grace and subtle predation. Kerygmachela kierkegaardi was a small lobopodian, measuring roughly 10 centimeters in length. Yet it embodied a remarkable synthesis of primitive and specialized traits. Fossilized specimens from the Sirius Passet and Burgess Shale reveal a creature whose morphology, locomotion, and ecological role provide unique insight into the experimentation of early animal life. Kerygmachela's body was elongated and soft, segmented into distinct regions, with paired lateral flaps running along much of its trunk. These flaps, flexible and gill-bearing, enabled swimming through gentle undulations and allowed subtle hovering just above the seafloor. Unlike more aggressive predators, Kerygmachela relied on stealth and environmental awareness, using its flaps for precise movement rather than rapid pursuit. Fossil impressions show the arrangement and articulation of these flaps, highlighting an evolutionary solution that balanced efficiency with maneuverability in a densely populated Cambrian ecosystem. Its head was equipped with a simple mouth and a pair of anterior appendages that functioned as sensory and grasping organs. These appendages were not heavily armored or spined, but allowed Kerygmachela to manipulate small prey, probe sediment, and detect environmental cues. Fossils indicate a degree of flexibility that would have been advantageous in foraging within complex reef structures or between microbial mats. This dual functionality illustrates a predatory strategy focused on opportunistic feeding, where precision and environmental awareness were paramount. One of Kerygmachela's most striking features was its tail region, which tapered into a pointed structure that likely aided in stabilization and steering. Fossil evidence suggests that subtle contractions of body segments, coordinated with lateral flap movement, allowed controlled hovering and precise positional adjustments. In combination with the anterior appendages, this allowed Kerygmacula to ambush small worms or arthropod larvae while remaining concealed, highlighting a strategy of patience and calculated predation rather than speed or brute force. Sensory adaptations were essential for survival. While it lacked the stalked compound eyes of many anomalocaridids, Kerygmacula likely possessed simple visual structures capable of detecting light and shadow, complemented by mechanosensory inputs from the body and appendages. This combination allowed it to detect prey and predators alike, navigating the seafloor environment with careful precision. Fossils showing preserved impressions of appendages and body surfaces suggest sensitivity to water currents and tactile stimuli, crucial for a creature relying on stealth and timing. Ecologically, Kerygmacula occupied a niche that bridged the gap between scavenging and active predation. It could feed on soft-bodied worms, small arthropod larvae, and organic detritus, exploiting resources unavailable to larger or less flexible predators. Its dual strategy allowed it to remain adaptable in changing environments, navigating both open seafloor and complex microhabitats. Fossil evidence indicates patterns of foraging and habitat use, providing a window into the subtle ecological interactions that shaped Cambrian communities. Evolutionarily, Kerygmacula represents an intermediate form, linking lobopodians to more derived panarthropods. Its body plan combines soft, segmented anatomy with functional lateral flaps, flexible appendages, and a tapered tail, demonstrating early experimentation with locomotion, sensory input, and predation. Fossils preserve fine anatomical details, revealing how evolutionary pressures produce solutions that combined efficiency, flexibility, and ecological versatility. Fossil evidence also illuminates Kerygmacula's locomotion and feeding strategies. Impressions of lateral flaps show coordinated undulation patterns, while appendage positions suggest deliberate probing and grasping motions. The integration of sensory input, body movement, and predatory function indicates an organism capable of executing complex behaviors despite its small size. This coordination between structure and behavior highlights the sophistication of Cambrian lobopodians, challenging assumptions about the simplicity of early animals. Despite its ingenuity, Kerygmacula disappeared from the fossil record, likely due to ecological shifts and competition from more specialized predators. Yet its legacy endures, providing insight into the diversity of survival strategies during the Cambrian explosion. Fossils capture a creature that combined precision, patience, and environmental awareness to thrive in a world dominated by both predators and prey. Through the study of Kerygmacula, scientists gain an understanding of how subtle predation and ambush strategies contributed to early ecosystem dynamics. Its morphology and behavior illustrate that evolutionary success did not always rely on size or speed. Adaptability, sensory acuity, and precision were equally critical. Fossils provide not only structural detail, but context for understanding ecological interactions, offering a glimpse into the delicate balance of Cambrian life. Kerygmacula reminds us that evolution is an experiment in form, function, and behavior. Its elongated body, lateral flaps, flexible appendages, and tapered tail illustrate a predator that moved with intention, striking at prey when opportunity arose and navigating the complex seafloor with care. In the currents above microbial mats and reef structures, Kerygmacula hunted, foraged, and survived, a testament to the subtle ingenuity of early animal evolution, preserved in stone for millions of years to inspire and challenge our understanding of life's early experimentation.